So everyone cool with that? Okay, good. I just wanted to double check with you guys because I was getting concerned about that. Okay. So let's go into presentations. Let me stop my share so you guys can start. Let me just log in. Who wants to go first?
Can I go first?
Sure.
Okay. Hold on. I don't know why the. Okay. Can you guys see it?
Yes.
Okay, so I had question 2, and the question was, a father with myotonic dystrophy has 3 daughters who are all carriers of the mutant allele, and 2 sons who are unaffected non-carriers. The 3 daughters have 6 sons, of which 4 are affected and 2 are not, and 4 daughters, of which 2 are carriers and 2 are not. From this description, what type of mutation is probably responsible for myotonic dystrophy? So when I read this question, I just reread it because that was a lot. So I started off with the first sentence and I just reread that. So it said, a father with myotonic dystrophy has 3 daughters who are all carriers of the mutant allele. So we know this is just passed down by the dad and not the mom. So that means it's not, that means it's not autosomal recessive. If it was autosomal recessive, that means it would be passed down by both the mom and dad. So we can already eliminate B. And then by the first sentence again, it was not passed down to the sons. And the sons have the Y chromosome like from the dad. But it wasn't Y linked because it only got to the daughters. So we can eliminate Y. So we don't have B or D. So it's just between A and C. So now we just have to figure out, is it dominant or recessive? So for it to be dominant or X-linked dominant, the daughters will have to show the myotonic dystrophy, which they don't. If they were, they will have the actual disease, but they're just carriers of it. So this means they're recessive. So the answer is C, X-linked recessive. Any questions? No?
I like how you explained it. It's very good.
Thank you. Me too.
Good job.
Thank you. So essentially, that's how you guys should break down these questions, okay? These questions, like with extensions of Mendelian genetics or other questions, they can get wordy if you haven't noticed. So break it down to the parts. If you have to write it out, like that's how I do it. Sometimes I do pedigrees to see what's going on. So just make sure you find a way that you are comfortable with to actually break down what the question is asking you, just like Yvonne did. Okay? Great. Okay. Who wants to go next?
I could go next.
Okay, okay.
Okay. Okay, so my name's Kiara Paris, and I had question number 3. And my question said, a female is diagnosed with hemophilia, a condition caused by an X-linked recessive gene. While checking her medical history, the doctor will most likely discover that A, her father had hemophilia and her mother was normal. B, all her siblings also have hemophilia. C, there is no family history of hemophilia, or D, her mother had hemophilia and her father was normal. So in my research, since males have only one X chromosome, that this causes them to be more likely to carry recessive X-linked disorders. So when a female inherits and expresses an X-linked recessive disorder, it's usually her father that has the disease. So this causes my answer to be A, her father had hemophilia and her mother was normal.
Yep.
That's it. Okay, that's all I have.
So one of the other things that I don't know if you said this, but with that question itself, the mother could be a carrier but not show anything, right?
Okay, yeah.
Yep.
Forgot to mention that.
Does that make sense?
Yes.
Because if it was just one allele, it would have been dominant, right?
Mm-hmm.
Good. Okay.
Okay, thank you.
Yep. Does that make sense? Okay. Loisha?
Yes.
You are presenting today, right?
Yeah.
Okay.
Can you guys see?
Nope. Oh, no. Yes. Well, it's coming. Are you sharing it now? Loisha? Okay, what's going on?
Okay.
I can't tell if you're frozen up or okay.
Hello?
Yeah.
Yes, I am.
Is your internet going down or something?
I think that connection is not good.
Okay. Did you at least upload the PowerPoint to Blackboard? Yes. Let me just double check. Okay, you do have an uploaded version. Okay. Um, so let me see if I can share it for you guys so you can actually cover that question too. Okay. Let me just open that guy up. Yeah, yeah, yeah. Okay. Can everyone see the PowerPoint itself?
Yes.
Okay. Loisha, are you able to speak or do I need to go over this?
Wait, how come my screen is dark?
Hmm?
Her screen is like dark on ours. I don't know if she's here.
Yeah, my screen is dark too.
It's dark.
Like it doesn't say her name, so it's just like an empty slot. Like
Oh. Okay. So essentially, the answer to this is D because we're asking incomplete dominance, right? So when you have flowers of color traits exhibiting here, you have so you have a red one and a white one, right? So in incomplete dominance, it's a 1 to 2 to 1 ratio, right? The heterozygous ends up showing an intermediate color, right? So that's how you end up with D. Does that make sense?
kinda.
So this is kind of her way of putting it. This was, so we have the, so R1 is red, R2 is white. So this would be like, so when you cross it together with, when you have the second set, so the question is, so the progeny of that guy shows incomplete dominance. That means there's an intermediate going on, right? So when you have a red flower and a white flower, you would have 1/4 of them, the F2 population. So at that point, you know it's heterozygous. So the parents themselves are homozygous for their trait. When you come to the F1 state, the first generation of the progeny, you end up seeing incomplete dominance completely. So that means they're heterozygous and they end up with an intermediate color, in this case, pink. So when you have a monohybrid cross, since we're only looking at one gene, this tells you that you have 1/4 of those guys are red, 1/4 of those are white, and then you have two types of heterozygous, right? Like one from the other, depending on which one you get. So in this case, they would be half pink. Does that make sense? Okay.
Yes.
Can you guys see my PowerPoint?
Yes.
Okay, just want to make sure. Yep. Oh, yep. Okay. So today we're going to talk about two topics that are coming up in lecture. The first one will be chromosome mapping, and the other one would be sex chromosomes and determination of sex, okay? So before we start off, we're gonna talk about crossing over. What was crossing over? Do you guys remember?
What's it when like the chromosomes would like cross over and it would make them like it would just exchange DNA between the 2
So essentially the definition is the exchange of genes between two homologous chromosomes, okay? So this occurs in prophase 1. It pairs homologous chromosomes and fuse together like this and exchange their DNA. This ends up giving more genetic variation, and it is also called genetic recombination, okay? And genes end up independently assorting in this case. So this ends up allowing more genetic variability. Okay. So one of the first things when we talk about chromosome mapping is the idea of linkage. So linkage ends up involving genes that sit close together on a chromosome, making them likely to be inherited together. So in this case, we have a chromosome. We have two genes that are linked, which means that they're close enough that they can be inherited together. Okay? But not all genes on the same chromosome can be linked, because like there is a scenario where if it's farther away from each other, so much to a point, you can see recombination or crossing over occurring between those two genes. So when that crossing over occurs, it can separate out and kind of make it more of a difference, okay? And then we have genes on separate chromosomes. They are not linked. They are on separate chromosomes for a reason, okay? So examples of linkages, that kind of thing, okay? So one of the things that linkage affects is it affects independent assortment. When they're linked, they cannot independently assort. In this case, so when you're thinking independent assortment, always think multiple genes, okay? So that's one of the things. So it won't end up separating out. So in this case, we have a parent. So we have the parents. They come together and they kind of recombine. So not recombine. So with these parents, we have these gametes, right? So two of them look like the parents. Those are called the parental groups. Then we have two that are differentiating that look different from the parents themselves. Those are called the recombinant ones, okay? So in linkage without crossing over, so when there's no crossing over happening, you'll only see the parental genotypes, right? Whereas with linkage with crossing over, you'll see all four of them, okay? Does that make sense? Yes, no, maybe. Where am I losing you guys?
Can you go over the last part?
So linkage with crossing over? This part?
Last two actually, without and with.
Okay, so we know what linkage is, we know what crossing over is, right?
Yeah.
Okay.
Yeah.
So when there is no crossing over between the two genes, when they're linked, you only, without crossing over, you only see the parents, right? Because you're not recombining or crossing over or anything. You're just staying there, nothing happening, nothing fun, just being like, hey there. And you just kind of go with, you only see the parental genotypes in your offspring gametes. Does that make sense?
So
Yeah.
basically, when it's not crossing over, you just see the parental ones.
So when you have linked genes with no crossover, it's just parental genotypes. When you have linkage with crossing over, you end up with recombinant DNA, gametes. So if you have, if you don't have crossing over, you just see these two in equal proportions. So you see half and half of those guys. Whereas if you have linkage with crossing over, you see all four in different numbers. Does that make sense? Yes, Yvonne.
Yeah,
So
and
does
it
this
makes
go with
Yeah.
Mendelian's ideas or go against his ideas?
So it's going, this is all going against Mendelian's, Mendel's postulate of independent assortment. That answer your question? Okay. Any other questions? Okay. Linkage affects expected ratios because it violates which of Mendel's postulates? D, right? Independently assorting.
Yeah.
Good.
Yeah.
So again, they're close enough that they cannot, they end up traveling as a unit. So independent assortment ends up being violated. In this case, the other choices would end up dealing with a single gene. So that is your answer for that. So the next thing about gene mapping is that you can use it to find the location of a gene by looking at how often different genes get inherited. So in this case, you can, so linked genes end up getting inherited more frequently together. So in this case, recombination is less likely to occur between linked genes. Less than 50% recombination means that the genes are linked, okay? So if you see any recombination frequency, so recombination frequency is just the number of, is the percent of recombinant progeny. Does that make sense? So when you have that frequency less than 50%, that means they are linked. When you see that, that just says that you have, you wouldn't have that much recombination going on. Whereas if you see it over 50%, you'll end up with independently assorting genes. And they are not linked, okay? Does that make sense? And then gene mapping itself can allow you to determine the order of different genes. So in this case, recombination frequency is actually equal to the distance between the genes themselves. So in this case, you have one map unit equaling 1% recombination. So the recombination frequency, 1% of that equals one map unit. So an example for that would be this guy. We have, so we have this set where we have 49 meioses with no crossing over. You have 49 of each of the parent ones, right? Then you have one set that is actually with a single crossover, nothing too fancy, just so you have two parental ones or non-recombinant ones, same here. And then you have the two recombinant ones because you have the changes here with that single crossover. Then one of the things you would, to calculate this guy, you would do number of recombinant progeny. So this one plus this one. So we have 1 plus 1 over the entire progeny number. Okay, so the summation of this guy, all of these together. So that would equal 2 over 200 times 100, which will give you 1%. Does that make sense? So that's essentially the frequency of recombination in this guy. Does that somewhat make sense? So let's try a couple of these questions and then if you guys have any questions. So two genes that are 25 map units apart are expected to show what's happening there. What do you guys think?
Is it B?
Wait, which did you say?
B?
B? Yes.
Yes.
So essentially, we know that it is less, when it's less than 50%, you know that it is involving 25% recombination, which means that you'll have that. When it's over 50%, you'll know it's independently assorted, okay? Does that make sense?
So does the number of like the map units correlate with the recombination frequency, or is that just a coincidence in this case?
So one map unit or the distance is equal to the recombination frequency. So one map unit equals 1% recombination frequency. Okay. Any other questions?
Can you please go back to the question? I want to see something real quick, please.
Everything okay?
Yeah, thank you.
Okay. We don't have any other questions. Let's try this guy. Assume that a cross is made between this genotype and this genotype plant, and the offspring occur in the following numbers. So we have 106 of this guy, 48 of this guy, 52 of this guy, 94 of this guy. These results are consistent with the following circumstance. Okay, wordiness. Okay, so let's go through this problem together. So what do we know? We know we have our parent genotypes. In the offspring, we have this many of that genotype itself, right? Then we have our recombinant genotypes, which end up with, so we know that these are recombinant ones because they look different from the parent genotypes, right? So you take those we were like, okay, let's think about the recombination frequency or the map units in this case as well. So we know that the total number of recombinant progeny is 48 plus 52, which gives us 100. Then the total number of progeny is all of these guys summed together, right? So when you do these guys, you get 300. So in this case, the recombination frequency or percent would end up 33% because it's 100 over 300 because it's total number of recombinants over the total number of progeny times 100. So it's the percent of recombinant genotypes out of the total, right? So that's how you get 33%. Then one of the questions we ask is that so we know that 1% recombination is equal to 1 map unit. So in this case, you would see that 33% recombination ends up with 33 map units. Does that make sense? So in this case, the answer ends up being D. Does that make sense? Okay. So let's try out this question. 3 genes on the Drosophila X chromosome are examined pairwise. It is determined that they have the following recombination rates. So yellow and white, 0.5. White and miniature is 34.5%. Yellow and miniature have 35.4%. Which 2 genes are physically closest together? What do you guys think? Okay.
Would it be A?
Yes. So what do we know? We know these guys are the recombination frequencies. We know that 1% recombination equals 1 map unit. And then that way we know that the distances between these genes are equivalent. So we get these guys. And since yellow and white have the shortest map unit distance, the answer is A. Does that make sense?
Yes.
Okay. So one of the next things you can do with chromosome mapping is actually finding the order of these genes. So this is a question related to that. A trihybrid cross produces the following recombination frequencies. V and CV equals 6.4 map units. V and CT equals 13.2 map units. And then CV and CT end up with 19.6 map units. What is the order? So to start off with this, you take the first step is the longest map unit you have. The longer map unit means they're further apart, right? So you know that CV and CT are the outer edges in this case. So they are going to be on the outside. Then you have V, right? Then you got, so what you would look at is the next part is which is remaining out of the 3. So in this case, we have V. So V can either go in the middle, over here, or over here, right? So in this case, what we know is that neither of these numbers are higher than this guy. So you can definitely cancel out anything that is on the outside. So that's how you would come up with V being in the middle, okay? So in this case, or another way is that these two kind of add up to 19.6. So they're kind of in the middle, right? So This is supposed to be CT. Sorry about that. But C, B, and V have 6.4. B and CT have 13.2. So that's how you get that order. CV, V, CT. That's how choice A comes into play. Does that make sense?
I have a question.
Yes.
Will the test ask us for the stuff on the left or the stuff on the right?
Wait, say that again?
Will the test want us to know exactly how it looks like you drew it on the right? Or is it
Well,
just looking for the basic order like on the left?
So it's essentially just asking the order of them. So essentially you would have to figure out where the one that is so I think it did say that he did want to know 3-point mapping. So in this case it'll just involve 3 different genes. These are the 3 different genes. There he will ask what order would it be. Does that make sense? Or did I misunderstand your question?
So it won't take the step 2 picture on the right. It's not going to put V closer to the CV and then give us like 2 of those pictures and tell us which one we have to look at and see which one is the correct one.
They wouldn't, I don't think Dr. Asuna would put in pictures like this. I just wanted to show an example as to how you would kind of figure it out.
Okay.
Does that make sense? But it would just probably be one of these guys, like this kind of question. I'm not entirely sure because I don't see the exam until you guys do, so. Does that make sense? Let's try out this question then. So what's the order of these guys with the 4 different loci? So where do we start?
Would you want to do the longest one first?
Mm-hmm.
And then the shortest one. So is it going to be like RNL first and then RNQ last and the rest in the middle?
So R and L are definitely at the ends. And then, so you can adjust it by looking at it, right? So you know that from R and Q, it's only 15. Q and L have 40. So you kind of realize it's closer to R, farther from L, right? And then you have W, which is equidistant between Q and L, right? So that's how you kind of get that order. It can be in backwards, forwards, however way you want to put it. But does that make sense? Okay. Any questions before we move on to sex determination and chromosomes? Okay. So these are the sex chromosome compositions of common model organisms. The one we're familiar with is fruit flies and mice and humans themselves, the XXXY mode, which is also called the ligase mode. And so C. elegans, they have XX and XO, which means that they don't have a second chromosome for that. That's called the protonor mode. I've seen Dr. Usuna give questions on this, so make sure you guys can remember like a example of a protonor mode or or the ligase mode. So one of the things that comes up with C. elegans, I like to talk about it because I work in a C. elegans lab. So this is so this is Lena, our I guess mascot in a sense. So this little beauty of a C. elegans does not like it when people forget what this means. So remember it forever, or at least until your exam. You don't want Lena upset, okay?
Lena seems chill though.
She is chill until people forget what Codinor mode is. Then she'll be sad. Okay? So don't forget it. So the main components of the Y chromosome so there are three main regions that we need to know. So the first one is pseudo-autosomal regions, or PAR. The second one is the sex-determining region and the male-specific region of the Y chromosome. So this male-specific area, no recombination. That's pretty much what you need to know with that. With PAR, that's the part where it shares homology with the X chromosome. So that's how they end up aligning as homologous chromosomes. And then with the SRY, which is also actually part of the male specific region, that's where you have something called, it encodes a protein called TDF, or testes determining factor. Does that make sense?
Yeah.
So what does the PAR do?
Wait, say that again?
What does the PAR do?
Okay, so essentially the PAR is just allowing it to be homologous with the X chromosome. And it's important when it comes to crossing over and all that fun stuff. The male specific region does not recombine. So to have it line up with the X chromosome, that's where the PAR comes into play. Does that make sense? Okay. So what did the studies of these chromosomal abnormalities show about the Y chromosome? So you have all of these X's, right? They all end up females. But then to all of this, if you just add one Y, it ends up male. Oh, okay. So kind of gave away the answer to that, but essentially the presence of the Y chromosome is what shows or determines maleness, right? That's what it shows in humans, okay? So the next thing would be talking about bar bodies. Bar bodies are also known as sex chromatin bodies. These guys are darkly stained bodies in the female interphase cells. And essentially what you need to know about this guy is that they are inactive X chromosomes. So if you have more than one X chromosome going on, one will remain active and then every other X chromosome involved gets inactivated. Does that make sense? So this is a bar body. That's just a picture there. So what do you guys think with this guy? How many bar bodies would you see with Klinefelter syndrome, which has this genotype?
One.
Yep. What about Turner syndrome?
None.
Yep. Good job. What about this?
None.
with this?
Two.
Yeah.
2, right? Because you have more than one chromosome, X chromosome.
It's just the number of extra chromosomes.
Mhm. So you have one remains active, and then any other X chromosomes, they are inactive. So those inactive X chromosomes end up being bar bodies because they're inactive.
I have a question.
Yes.
So that guy with the XXY, is he gonna be a male too?
Yes. That is a male. So what about for this
Triple
guy?
X is a female,
Huh?
and the triple X is a female.
Wait. This one?
And
Yes.
the triple X is female.
Yes. The triple X is female because there's no Y chromosome.
This way.
Okay. What about this guy?
3.
3,
Yep. This one?
Aren't those diseases?
0,
Yes. They are
2.
diseases.
0.
1.
Yep. 0. Because
These,
it's, oh, what happened?
Why wouldn't it be 1?
Because the X chromosome, if it's just one X chromosome, that's the active one. If you have more than one, that's when you end up with. So one X chromosome is always active. Then the rest end up, if you have multiple ones here, X chromosomes, those guys end up being inactive, right? So this first one ends up active, the remaining ones end up inactive, which makes them bar bodies. Does that make sense? Okay.
Yes.
So the mechanism for inactivation here involves what's called an X inactivation center. So it's essentially active only on the, so it's pairing up the X chromosomes. It's only active on the X chromosome that is inactivated. It contains several genes that are important for the inactivation. So in this case, we have 3 different RNAs that play a role in this. So one is cis gene, which is critical. Deletion of that gene ends up with no inactivation of the X chromosome. Then you see that it associates with the X chromosome and carries the genes that produce the inactivation stuff. Then T6 gene is just the antisense RNA partner for Xs. And X8 gene is just transcribing a non-coding RNA. And all of this comes together and they transcribe everything and they're like, okay, like inactivating time, when it's activated, it's like boom, 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 covering all the genes and be like, no, you are not transcribing me, right? So that's essentially what's involved with the mechanism, right? Are we okay with this?
Yeah.
So Lyon hypothesis involves that it's stating that inactivation of the X chromosome is completely random. And it starts occurring at the blastocyst stage, right? And all the descendant cells end up having the same pattern of X inactivation. So in this case, an example for this that demonstrates Lyon's hypothesis is calico cats and tortoiseshell cats and their fur patterns, right? So essentially, it's just saying that it's random and it's not complete, okay? So patches of expression depending on which chromosome is inactive. So in this case, you can have a black cat with orange patches if it's, so this here is just showing that the inactive chromosome. So in this case, you have black and orange going on, but then like it depends on which one is inactive and its expression is differing between them. Does that make sense? Okay. So cat breeders are aware that kittens expressing the X-linked calico pattern and tortoise shell pattern are almost invariably females. Why are they certain?
I'm sorry for cutting you. Can you please go back to the last slide? I didn't quite get it.
Okay, so essentially it's just saying that the inactivation is random. And in certain cases with like this mosaic pattern comes out because inactivation is not entirely complete. So some genes can actually come out and express themselves. That's how this mosaic pattern comes in the cats. Does that make sense?
But wouldn't that make it incomplete dominance?
So what I'm thinking is that it is, so these traits are on the X chromosome. So there are different rules with the sex chromosomes going on. Does that make sense? So
No,
huh?
really, I don't get it.
Okay. So, okay, let me go to one of the other slides. So this question kind of answers that part. So the reason why this happens is that since the what's the word? Inactivation of the X chromosomes only occur at this stage. There are this many stages before that, right? So it's just saying that with these cats, these genes are still kind of expressed because it's random, and these guys can, since it's only starting at this stage, it only ends up with some of the genes actually escaping inactivation and it's not being fully covered. Does that make sense? Yes, no, maybe.
Maybe.
Okay. I'm pretty sure the quiz and so since we're kind of a little bit over time now, is 8:27. I don't want to hold you guys too late. We'll pick it up from Lyon's hypothesis next week. Okay. With this so for the quiz, your questions will be based on chromosome mapping. I'm pretty sure the presentation questions are also from that topic too. So if you guys have any questions, let me know. For attendance, just write the word here in the chat. I'll try and find a better video or something that describes Lyon's hypothesis. Because for one thing, it's not incomplete dominance because it's more so with X-linked or X inactivation. That's specific to the sex chromosomes. So it kind of goes there. So this part here, it's just saying that it kind of, so these are the genes that can be expressed. That's why it's showing up in the cat. If that made any sense. Yes, no, maybe so. Okay. So essentially they're certain of this because they have two X chromosomes randomly inactivated. Males don't show the mosaic pattern because they only have one of the two X-linked alleles. But yeah, that's all I have for today. Make sure you guys write the word here in the chat. You guys have not done that already.
Thank you, Nimi.
Thank you. Have a good one.
Thank you.
Thank you. Have a good night.
Have a good night, guys.